Hi guys, and welcome to this chapter 18 PowerPoint presentation recorded in advance. Thanks for listening to me in a recorded way. So this chapter is on the urinary system and fluid balance. Chapter 19 will be on reproductive, and then chapter 20 will be on kind of development and hereditary. And these three chapters will be what makes up your final lecture exam. So your urinary system is your major excretory system of the body. Some organs in other systems also eliminate wastes, but your kidneys are what do the majority of your excretory or excretion of wastes that your body needs to get rid of. And it does that by filtering the blood. And those other organs in the other systems are not able to compensate for excreting body wastes if your kidneys ever fail. So this is what makes up your urinary system. You have two kidneys connected to ureters, connected to a urinary bladder, which stores the urine, and the urethra is what takes urine out of the body. So your kidneys will filter blood to create urine, the kind of the liquid carrier of all the waste that needs to leave the body. The urine will travel through the two ureters and be stored in the urinary bladder until a reflex occurs and it allows the urine to be emptied through the urethra. So here are the functions of your urinary system: excretion of wastes. It also helps to regulate blood volume and blood pressure. It regulates blood solute concentration because it can retain or absorb solutes back into the bloodstream, or it can secrete excess solutes into the urine to get rid of them. The urinary system helps to regulate extracellular fluid pH. It regulates red blood cell synthesis because erythropoietin is secreted by the kidneys when you need more red blood cells. And it also regulates vitamin D synthesis. So these are the components of the urinary system that were shown in that previous picture that you should know well. Here is just another look at the urinary system. We'll go over this in lab this week. The two kidneys connected to the ureters, and then bringing it down to the bladder, and then the urine will be out of the urethra. Here are kidney characteristics. They're bilateral retroperitoneal, meaning they lie on the back side of the abdominal cavity. They're bean-shaped. They weigh about 5 ounces, which is about a bar of a soap for the size of your fist. And they're located in the back between the T12 and L3 vertebrae. Each kidney has a renal capsule, which is a really tough connective tissue around each kidney, which will protect and act as a barrier. The hilum is the medial indentation, which is where the renal artery, veins, nerves, and ureters will enter and exit the kidney. The renal sinus is an area within the kidney that contains the renal pelvis. We'll talk about that, all the blood vessels and fat. The renal cortex will be the outer layer of the kidney under that fibrous capsule, and the renal medulla is the inner portion. And again, when we get to pictures, this will make a lot more sense. Renal pyramids are kind of at the junction between the cortex and the medulla. They lie kind of within the medulla. We'll see how that works. The calyx is the tip of the pyramids where urine that is formed is kind of funneled out. The renal pelvis is where all of the calyxes join together, kind of this big funneling system, which will eventually narrow to form the ureter to take that formed urine out of the kidney. So here we have a longitudinal section of the kidney, and if you zoom in, you can see the renal capsule, and then underneath the renal capsule is the lighter colored renal cortex layer. And then below that will be the darker colored renal medulla layer that contains these renal pyramids, which look kind of like these pizza slices. Each renal pyramid is connected to a calyx, and all these calyxes connected to these renal pyramids will join to form the renal pelvis, which again is kind of a big collection funneling system of urine that will eventually bring urine out of the kidney through the ureter. The nephron then is what we call the functional unit of the kidney. Each kidney has over 1 million nephrons. There's 2 types. 15% are juxtamedullary. The nephron includes the renal corpuscle, the proximal tubule, the loop of Henle, the distal tubule, and the collecting duct. The renal corpuscle itself is a structure that contains a Bowman's capsule and a glomerulus. The Bowman's capsule is an enlarged end of a nephron that opens into the proximal convoluted tubule. It contains specialized cells called podocytes, which kind of surround glomerular capillaries. And then the glomerulus itself contains capillaries wrapped around it. So here we have the renal corpuscle and this glomerulus structure, which we call the filtration membrane. So the idea is that blood is taken into this glomerular capsule, also called the renal corpuscle. And when blood is taken in through the renal corpuscle, it gets filtered out, and all that filtrate then gets picked up by this Bowman's capsule and then travels through the proximal convoluted tubule. So that filtration membrane refers to the idea of blood and solutes being filtered out of the blood into the Bowman's capsule. And you can see a zoomed in look at the filtration membrane. So you see kind of these capillaries seen here with these podocyte cells with slits kind of between their feet. And what that means is when blood travels through the capillaries, all the solutes and whatever gets filtered through will travel through this filtration membrane and through these little slits of the podocytes. And this filtration membrane is important. Some things cannot get through it, like red blood cells. So you should never see blood in your urine. Other things can get through. Usually proteins are too big to get through. So if you see proteins in your urine, that usually means there's a problem at this filtration membrane. So the filtration membrane is kind of our first part of the nephron components. It's in the renal corpuscle. It includes your glomerular capillaries, podocytes, the basement membrane. Filtrate is the name that we give for the fluid that passes across the filtration membrane and then travels into the proximal convoluted tubule, down and up the loop of Henle. During this loop, water and solutes will always pass through thin walls by diffusion, either into or out of the filtrate. The distal tube is the next structure in the nephron. It's the structure between the loop of Henle and the collecting duct. And then the collecting duct is what empties into all the calyxes. It carries fluid from the cortex through the medulla and eventually to the calyxes, to the renal pelvis to get that waste, that urine, out of the kidney. So here's a look at that nephron. We have the renal corpuscle, and then we have an inside look at the renal corpuscle showing the glomerulus and the Bowman's capsule. So the filtrate that is filtered through the blood will travel through the proximal convoluted tubule first, then through the loop of Henle. And you can see that you have different descending and ascending limbs of the loop of Henle. It will travel through the distal convoluted tubule and then down the collecting duct, where it kind of gets funneled to a calyx, and it'll eventually leave the nephron. So you should know this order of flow of the filtrate through the nephron, starting at the renal corpuscle, and then ending in the collecting duct, and then traveling through the papillary duct into the calyx. So you should know the specific order that filtrate goes through the nephron. The blood flow through the kidney is shown here. I will provide you guys a list of study tips. I don't know if you need to know specifically this blood flow scheme, but I will let you know that. Remember, the whole role of your kidneys is to filter blood and create urine from the waste in your blood. So it has a very intricate blood flow system through the kidney that basically starts with the renal artery, travels through the lobes, out into the peripheries of the kidney, through the lobulars of the kidney, into the afferent arteriole. And the afferent arteriole is what will go into the glomerulus, which starts the nephron system of filtration. And remember, you have about 1 million nephrons, so you'll have about 1 million glomerulus. Then the efferent arteriole brings that blood out that's been filtered from the glomerulus. And then we go into our peritubular capillaries, the vasa recta, the interlobular vein, arcuate vein, and interlobar vein. So this just shows kind of that blood flow through the kidney going in number order. And this is really interesting to look at because as we kind of zoom in, especially on the picture on the right, as we look at the afferent arteriole, bringing blood into the glomerulus and then out as the efferent arteriole. That efferent arteriole makes different types of capillaries, the peritubular capillaries that surround the convoluted tubules, and then the vasorecta capillaries that surround the loop of Henle. And those capillaries are important because this is where substances, solutes, water will be absorbed back into the body from the filtrate that was formed, and it goes right back into these capillaries and then back into the bloodstream. So here is the kind of basic simple processes of urine formation filtration, which we talked about occurring in the renal corpuscle, reabsorption, which involves removing substances from the filtrate, placing them back into the blood, and then secretion, which involves taking substances from the blood at the nephron area, other than the renal corpuscle, and putting it back into the nephron tubule. So here are the three main processes of urine formation, where exactly they occur throughout the nephron and what it means. Tubular reabsorption means things are being absorbed back into the bloodstream, and tubular secretion means things are being secreted, like solutes, water, back into the filtrate. So filtration, first of all, is the movement of water, ions, small molecules through the filtration membrane into the Bowman's capsule. About 19% of plasma will eventually become filtrate, and 180 liters of filtrate are produced by the nephrons each day, which is a huge amount of fluid being produced by your kidneys. Only about 1% of the filtrate will actually become urine, though. So about 1.8 liters of urine is formed every day, while the rest is reabsorbed back into the water, the rest of that filtrate. Like I talked about before, only molecules that are small enough to pass through the filtration membrane will, and the formation of filtrate depends on the filtration pressure, or the pressure of blood, bringing blood into the Bowman's capsule. The filtration pressure forces fluid across that filtration membrane, and filtration pressure is always influenced by blood pressure. And this just shows how the influence of blood pressure, we have different types of capsular pressure, osmotic pressure, colloid pressure. Basically, we get kind of a net pressure. Things are pushing back into the blood, but the biggest push is coming from the blood pressure, pushing the filtrate out. So we get this net pressure, net movement of solutes and filtrate out of the blood and into the capsular space. 99% of filtrate is reabsorbed and will re-enter circulation. The proximal tubule will be that primary site for reabsorption of solutes and water. The descending loop of Henle will work to concentrate the filtrate, and the reabsorption of water and solutes from the distal tubule and the collecting duct is usually controlled by hormones. So here we have reabsorption in the proximal convoluted tubule. So just showing that most solutes plus 65% of the filtrate volume are reabsorbed from the proximal convoluted tubule and enter back into the peritubular capillaries. So most of the reabsorption occurs in the proximal convoluted tubule. Then urine concentration occurs in the descending limb of the loop of Henle, and this is where water is reabsorbed, so that when water is reabsorbed back into the bloodstream, the filtrate will become more concentrated. The mechanism for this water reabsorption is osmosis. The renal medulla contains very concentrated interstitial fluid that has large amounts of sodium chloride and urea, and the wall of the thin segment of your descending limb is very highly permeable to water. So filtrate moving through the medulla containing highly concentrated interstitial fluid, water will be reabsorbed just naturally out of the nephron by osmosis. The water will then enter the vasorecta capillary system that surrounds the loop of Henle. The ascending limb of the loop of Henle will dilute the filtrate by removing excess solutes. The thin segment of the ascending limb is not permeable to water, but it's permeable to solutes. So consequently, solutes will always diffuse out of the nephron. So a lot goes on here in concentrating the urine. And basically in the loop of Henle, whether it's the descending or ascending limb, whether it's the thick or thin limb, different solutes can be absorbed or secreted back into the filtrate at different parts of that loop. And then you see here on the distal convoluted tubule and the collecting duct, usually the reabsorption of water is controlled by hormones. So reabsorption in the loop of Henle, again, is showing here. One easy way to think of this is that wherever water goes, usually solutes will follow. So if water is osmosing somewhere, usually solutes will follow. And here's reabsorption in the thick segment of the ascending limb, again showing in the thick segment, most solutes are reabsorbed because water is not able to get through. The distal convoluted tubule and collecting duct reabsorption, again, so in the distal convoluted tubule and collecting duct, water and solutes can be absorbed, but again, most of this is under control by different hormones. So urine production then, the idea of secreting the urine, tubular secretion will remove some substances from the blood, and these substances are usually byproducts of metabolism that becomes toxic in high concentration, or other drugs and molecules that are not normally produced by the body. So this is why urine tests will detect drugs in the body, because usually that's one thing your body does not want to reabsorb, it wants to get rid of. And tubular secretion occurs through either active or passive mechanisms. So again, this secretion means secreting substances into the urine and keeping it there so the body can get rid of it as a waste. Ammonia secretion is passive. Secretion of hydrogen, potassium, creatinine, histamine, and penicillin is by an active transport. These substances just mean they're actively transported into the nephron, and the secretion specifically of hydrogen ions will play a very important role in regulating the body fluid pH of the body. So urine concentration and volume regulation, we have three main hormonal mechanisms that are involved in regulating urine concentration and volume. The Renin-Angiotensin-Aldosterone system, ADH is antidiuretic hormone, and the Atrial natriuretic hormone. So the Renin-Angiotensin-Aldosterone mechanism, sometimes called the RAA system, basically your kidneys will secrete renin in response to low blood pressure and low blood volume in the body. And renin will act on angiotensinogen to produce angiotensin 1. Angiotensin 1 eventually gets converted to angiotensin 2. And angiotensin 2 directly causes vasoconstriction. Whenever you vasoconstrict your blood vessels, blood pressure increases. So it works to try to counteract a decrease in blood pressure by increasing it by vasoconstricting your blood vessels. Angiotensin 2 also acts on the adrenal cortex of your adrenal glands, which lie on top of your kidneys, to release aldosterone. And what aldosterone does is it increases the rate of active transport of sodium in the distal tubules and the collecting duct. And so what that will lead to is a decrease in the volume of water in the urine, again, in a sense to try to increase blood volume and blood pressure. So here we have aldosterone actions. So we have aldosterone actions specifically. Any decrease in blood pressure will kind of put into effect this renin-angiotensin-aldosterone system. And specifically, aldosterone will increase sodium and water reabsorption back into the body and bloodstream to try to result in an increased blood pressure. ADH, antidiuretic hormone, is secreted by your posterior pituitary gland. It's one of two. ADA acts on your kidneys, causing them to absorb more water to decrease urine volume, and the result is to maintain a normal blood volume and blood pressure. So this is showing ADH release from the posterior pituitary, and that tells your kidneys to increase water reabsorption, which will result in a decreased solute concentration and increased blood pressure. Atrial natriuretic hormone then is secreted from your cardiac muscle. So that's why we see atrial. Specifically, it's secreted by the cardiac muscle in the right atrium of the heart when blood pressure increases. So this is the only one that actually does the opposite. So ANH will act on your kidneys to decrease sodium reabsorption. Sodium ions will remain in the nephron to become urine, and the increased loss of sodium and water will water will try to reduce blood volume and blood pressure. So here we have atrial natriuretic hormone released from the heart. And again, it does the opposite of the other two. So if we have an increased blood pressure in the right atrium, it'll tell your kidneys to increase sodium excretion and increase water loss to try to decrease that blood pressure. So here we have homeostatic control of blood and urine volumes using these different hormones to keep your blood pressure and urine volumes consistent. So the ureters themselves, you have two of them. These are the small tubes that carry urine from the renal pelvis of the kidney to the bladder. The urinary bladder itself is in the pelvic cavity that helps to store urine. It can hold a few milliliters to a maximum of 1,000 milliliters. And then the urethra is the tube that exits the bladder and carries urine from the urinary bladder to the outside of the body. So here's just a look. Sometimes students get ureters and urethra confused. So if you could remember that you have two ureters connecting the kidneys to the bladder and then one urethra. One thing to note is you have different sphincters. So you have smooth muscle that makes up the urinary bladder. So the contraction and release of urine in the bladder is actually an involuntary action. But the external urethral sphincter, that is made up of skeletal muscle that you do control. So you do control when the urine is expelled. Urine movement is called the Micturition Reflex, which is activated by a stretch of the urinary bladder wall. It sends action potentials from the bladder to the spinal cord through pelvic nerves. Parasympathetic action causes your bladder to contract, and the stretching of the bladder will stimulate sensory neurons to inform the brain that a person needs to urinate. So this is the Micturition reflex. Again, it's just showing that stretching of the bladder that causes action potentials to be sent to the brain and spinal cord. Then it comes back to kind of contract and release the urine. But this external urethral sphincter is under voluntary control. So you are able to control when the urine leaves the urethra. Then the body fluid compartment. So intracellular fluid compartment includes the fluid inside all the cells of the body, and about 2/3 of all the water in the body is intracellular, or inside your cells. When we talk about extracellular fluid, that includes all the fluid outside the cells. So that will include interstitial fluid in your tissues, plasma, lymph, and any other special fluids like joint fluids, the fluid that makes up the joint cavity, synovial fluid, and the cerebral spinal fluid. The composition of your fluids intracellularly, you have lots of ions, potassium, magnesium, phosphate, sulfate, much higher concentration of ions inside cells versus extracellular, but it has a lower concentration of sodium, calcium, chloride, and the bicarbonate ion than does the extracellular fluid. So the exchange between extracellularly and intracellular is controlled by the cell membrane, if you remember. So the cell membrane is selectively permeable. Water can continually pass through the cell membranes, but ions dissolved in the water do not readily pass through. Water movement is always regulated or maintained by hydrostatic pressure differences and osmotic differences between the compartments, and osmosis controls the movement of water between the intracellular and extracellular spaces. So some regulation of the extracellular fluid is thirst, regulation, ion concentration regulation, for example, thirst. Water intake is controlled by a thirst center you have in your hypothalamus. And when the concentration of ions in the blood increases, like after you eat a really salty meal, it stimulates your thirst center to cause thirst. And when water is consumed, the concentrations of the blood ions decreases due to a dilution effect, and this will cause the sensation of thirst to decrease. So this just shows how increased blood concentration tells your thirst center in your hypothalamus to drink water. Ion concentration regulation, so regulating the concentrations of positively charged ions like sodium, potassium, and calcium is very important because these are involved in sodium potassium pumps. Calcium is involved in muscle contraction, action potentials, muscle contraction, and normal cell membrane permeability depend on the maintenance of a very narrow range of these concentrations. So they always need to be always in their normal range. Negatively charged ions like chloride will be secondarily regulated by mechanisms that control the positively charged ions, and the negatively charged ions are attracted to positive, so that when positive charged ions are transported, usually negative charged ions are moved with them. Sodium is naturally dominant extracellularly. If you remember that from the nervous system, you always have more sodium outside the cell. About 90 to 95% of the osmotic pressure of the extracellular fluid results from sodium ions and from the negative ions associated with them. Specific stimuli will control aldosterone secretion, will influence the reabsorption of sodium from nephrons of the kidneys and the total amount of sodium in the body fluids. And sodium ions can also be excreted in sweat, but not much. Potassium ions then, so in electrically excitable tissues like muscles and nerves, they're highly sensitive to slight changes in extracellular potassium concentration, and the extracellular concentration of potassium must be maintained within a very narrow range for these tissues to function normally. Aldosterone specifically plays a main role in regulating the concentration of potassium in the extracellular fluid. So here's the regulation of blood potassium levels. Knowing the specifics of this, I don't think you need to know, but I will give you some study tips before your final exam. Calcium also needs to be maintained. Increases and decreases in calcium can have dramatic effects, specifically on the electrical properties of excitable tissues like muscles and nerves. Parathyroid hormone and calcitonin are two hormones that are antagonists. They work in opposite. PTH secreted by your parathyroid glands increases extracellular calcium concentration, and calcitonin reduces blood calcium or tones down blood calcium concentration when it is too high. Phosphate and sulfate ions are absorbed by active transport in the kidneys. The rate of reabsorption is slow, so that if the concentration of these ions in the filtrate exceeds the nephron's ability to reabsorb them, the excess will just be excreted in the urine. And as long as the concentration of these two ions is low, nearly all of them will just be reabsorbed by active transport. Regulation of acid-base balance. So our bodies use a lot of buffers, which are specific chemicals that resist changes in pH of a solution. And buffers are important. They contain salts of weak acids or bases that combine with hydrogen. So meaning they will kind of control the levels of hydrogen ions to control the pH levels. And you can have three classes of buffers. They can be proteins, a phosphate buffer, or a bicarbonate buffer. The respiratory system is also involved in maintaining this acid-base imbalance because it can respond rapidly to changes in pH. So if you have an increased respiratory rate, this will raise your blood pH, meaning it will make it more alkalytic or more basic. What this means is if your respiratory rate increases and you're breathing a lot, that means that you're increasing the rate of carbon dioxide elimination from the body. And if you get rid of carbon dioxide too quickly, the pH level of the blood becomes more basic. If you have a reduced respiratory rate, so you're breathing very shallowly, this will reduce the pH of the blood fluid levels in the blood, making it more acidic. And that's due to the decreased rate of CO2 being eliminated. So carbon dioxide builds up in your blood, and that creates a more acidic blood. Kidneys are involved in acid-base regulation by secreting excess potassium or hydrogen ions into the urine, or and directly regulating pH of body fluids. So more hydrogen ions will be secreted if the pH is decreasing. So if your pH levels get to be too acidic, your kidneys will get rid of excess hydrogen ions, and less hydrogen ion secretion will occur if the pH is increasing. So this is the regulation of acid-base balance in the blood. Acidosis and alkalosis are two problems. So normally you want your blood, pH of the blood, always around 7.4. If it gets below 7.35, we have an imbalance of pH called acidosis. And there's just two types of acidosis based on what causes it, whether it's respiratory or metabolic, whether it's caused by breathing or your kidneys. And then alkalosis occurs when the pH of the blood increases above 7.45, so it becomes a little too basic. And the two main causes of alkalosis can be respiratory or metabolic as well. So thanks for listening to this recording, guys. I will also record chapter 19, which is reproductive, because we're one day behind. And then we will meet live for live normal class on Wednesday to go over chapter 20, which will be our final chapter. And then we're pretty much done with the semester. So keep up the great work, guys, and let's finish strong and have a great rest of your day.